नमस्कार, आदाब, सत श्री अकाल।
यह है BHU का हैदराबाद गेट। गेट का कोई नंबर होता है? अच्छा, बड़ी गेट हैदराबाद बड़ी गेट के माध्यम से हमने एंट्री मारा है। और ड्राइवर भैया को कहीं फ़ोन करने का तलब आ गया है तो बतिया रहा किसी से। और ये है W कुमार, रियल क्या नाम है? प्रवीण कुमार। ठीक है, तो ये BHU कर रहे हैं, अब इनकी शादी नहीं हुई है।
ये वाला
हमसे कई बार बोला है कि आप एक बार बोल दीजिए।
मैंने एक बार भी नहीं बोला है, झूठ बोलना है।
कि एक बार बोल दीजिएगा अपने ब्लॉग में।
एक बार भी नहीं बोला हूँ शादी के लिए।
ठीक है, तो ये जो है तो कैसा दूसरा साल है ना?
नहीं बताएंगे।
दूसरा साल है PhD का। JRF है, अच्छा खासा कमाता है। कितना कमाते हो? JRF में जितना मिलता है आप गूगल करके सर्च कर लीजिएगा, उतना कमाता है ये। बनारस में रहता है, अच्छा खासा कोठी है,
ढाबा।
और इसका एक बड़ा भाई है W भैया, आप सबको जानते होंगे, नंटी भैया को। वो भी यहीं से PhD कर रहा है। तो अभी हम पहुंच गए हैं, लेकिन हमारा जो लेक्चर है 11:30 बजे, अभी बढ़ जाए 10:30। मेरे पास 1 घंटे का समय, मैं सोचा थोड़ा समय W भैया हमको घुमा रहा है, हमको ले जा रहा पकौड़ी खिलाने, है ना?
हाँ।
समोसा
नहीं समोसा सुबह सुबह नहीं।
यहाँ पे क्या मिलेगा?
यहाँ पे हिंदू वाड़ा मिलेगा।
हिंदू वड़ा, वाह, नाम सुन के तो ऐसा लग रहा है कि वाह।
चाय ब्रेड।
चाय ब्रेड मिलेगा। तो यही यहाँ पे इंदिरा कैफ़े है।
के सामने हम लोग
हाँ।
छोटे मोटे।
अच्छा, ये जूलॉजी डिपार्टमेंट है। तो जूलॉजी डिपार्टमेंट इधर है, वो लिखा हुआ है इंस्टीट्यूट ऑफ साइंस, और उधर है सेंटर फॉर जेनेटिक डिसऑर्डर, जहाँ पे मुझे जाना है। क्या कहते हैं? ये क्या है? आलू
बाटी चोखा।
बाटी? ये बाटी चोखा है?
बाटी
बाप रे।
चाहिए?
नहीं चोखा नहीं चाहिए, बस ये बाटी खाएंगे। बाप रे, सवेरे सवेरे बाटी चोखा। मुस्कुराइए, आप बनारस में हैं। तो नाश्ता वास्ता करेंगे यहाँ होते तुम? लंच
मोस्टली करते नहीं हैं, फिर करना होता तो फिर टिफिन ले आते हैं।
लंच कहाँ करते हैं?
उधर आगे है
अच्छा, चाय वाय के लिए यहाँ पे आते हैं।
चाय छोटा मोटा नाश्ता।
अमेरिका में अगर आप चाहे तो एक बाटी लो, चाहे चोखा लो, पैसा पूरा देना पड़ेगा।
दोनों जाता
मतलब दोनों लेबर तो दोनों को प्राइस रहे। अगर तू सोच भाई तोके चोखा ना चाहे, सिर्फ बाटी चाहे, तो तोको तब तोके पूरा पैसा देख पड़े। लेकिन हमें मर्चा बहुत ज़्यादा बा। हाँ, पहली बार हम नाश्ता में बाटी खा रहे हैं। मुझे लगा कचौड़ी है।
कचौड़ी भी था।
हाँ?
कचौड़ी भी था।
कचौड़ी है तो कह रहे हैं,
बाटी बोले हैं।
नहीं कचौड़ी मैंने बोला। चलो कोई नहीं,
कचौड़ी दे दें।
नहीं बोल। तू नहीं खाता?
मुझे अभी नहीं खाना।
तू नहीं खाता?
अभी नहीं खाना।
सादा जीवन उच्च विचार। जूलॉजी फाइनल ईयर है आपका?
नहीं, फर्स्ट सेमेस्टर।
चलो बढ़िया सब्जेक्ट है, हम सब जूलॉजी वाले ही हैं। आप पूछ रहे हैं। सही है।
कितने बजे से?
11:30।
कहाँ पे?
ये सेंटर फॉर
जेनेटिक डिसऑर्डर।
क्या नाम है?
सेंटर फॉर जेनेटिक डिसऑर्डर।
सेंटर फॉर जेनेटिक डिसऑर्डर। 11:30। ठीक है। तो हमारे पास है W भैया। तो W भैया हमको अपना लैब दिखाने लेके ले जा रहे हैं। हमारे पास अभी 45 मिनट का समय है। मैं सोचा
मेरा लैब पहले देखेंगे कि बाद में आते?
अब वो अपने लैब में लिया जाता है ना।
हाँ, मतलब टॉक के बाद।
नहीं, अभी चल रहे हैं ना। हाँ, तो अभी हम जा रहे हैं इनका लैब देखने। और इनके यहाँ 200 पे काम है ना? 200 मक्खी 15-20 मिनट में हो जाए ना। धन्यवाद। ये हैं पवन दुबे सर जी, जिन्होंने हमको इनवाइट किया है अपने यहाँ टॉक देने के लिए।
सही है। तो मैंने देखा, फिर मुझे लगा कि सर हैं अभी आ रहे हैं, और अचानक संयोग बन गया। मैंने 2-3 मतलब एक हफ्ते पहले आपका वीडियो भी देखा कि आप आ रहे हैं करके।
Today I'm going to talk more about workshop kind of talk, and I'm going to more talk about how gene therapy work and how you can use for your research, how gene construction you can design, and what are tool and technology, especially genetic engineering tool that you can use for your research, and also for your career point of view. If you write and if you have an experience of genetic engineering, any kind of genetic engineering, if you write in your CV, if any person will see your CV, they will very happy to see such kind of experience. Okay, another thing I want to say before I start that I changed my PowerPoint in last moment, so I did not prepare much. So I will try to use multiple slide, multiple PowerPoint, multiple slides from multiple files. So try to combine together to make a complete story. Okay, so first thing first, who am I? I have done my PhD from CDRI Lucknow and master's degree from KMPT Kanpur. Here are my expertise. My expertise in how you can target the gene overexpression and knockdown in particular cell subtype, especially all the research are in vivo. And second thing, how can you use the CRISPR technology to gene overexpression and gene knockdown. And as you know that if you want to study the RNA level in particular cell subtype, how you going to study the RNA? For example, if you extract the RNA from the brain, particular brain punch, chunk of brain size, that chunk have all possible kind of cells. How you going to isolate the RNA only one kind of cells? That's expertise I have. And lastly, recently, like 5-6 years ago, I started the gene therapy lab, where I do the gene construction designing. I package the gene into the AAV and lentivirus, and I provide the service for the other users within university as well as outside the university. Okay, so and my hobby is I have a YouTube channel, and where I try to teach, especially the lab at home, science in layman style, so that any person can understand the science is not quite difficult to understand. People who never go to school, that person also, if he or she will see my video, they will understand what I'm talking about. Okay, so what is the gene therapy? Very simple. Just fix the bad gene with the good gene, and whatever technology you are using is called gene therapy. So why gene therapy required? So as we know that any of the protein in our cells come from the DNA, and DNA translated into mRNA translated into protein, and protein has certain function. If any kind of mutation or addition of any DNA over there, or deletion or overproduction or underproduction of this gene, at the result this protein might not work as it should be, right? If we fix the problem, if we fix this gene by any method, then protein will work properly, and the related disease also will be fixed. This is called gene therapy, right? So gene means plasmid. Basically, we deliver the plasmid in the tissue, whatever disease associated with that particular tissue. Next question is how you going to deliver this plasmid into the cells. For example, if you have any injection syringe that have a plasmid in it, you just directly inject to the heart or kidney. Our cells do not like any kind of foreign object to go into the cells, especially in vivo, even in vitro, right? In vivo, our body is designed in such a way, our defense mechanism is so high that it will not allow to enter any plasmid into the cell. Maybe 0.001% plasmid might go inside the cell, but rest of the plasmid will be waste. So your aim will be not fulfilled, right? The question is how you going to deliver the gene of interest into the cells. There is a many ways to deliver the gene into the cell. The way that I'm going to talk about is called viral based gene therapy. And I have expertise on delivering the gene of interest using viral based. One is adeno-associated virus, another is lentivirus. I will more talk about adeno-associated virus based gene therapy. But if you have any questions related to lentivirus, feel free to ask. Like I said before, I prepared the slide at the last moment, so I do not compile much enough. So feel free to ask any question. The question is why we are using the virus. If you think about virus in your, if you think of virus, first thing come in your mind is very infectious, right? For example, coronavirus. You know, the whole world is affected because of the infectious in nature. Virus is one of the organism, microorganism, parasite that has highly infectious in nature. You can take advantage of this. First is because of this has very highly binding affinity. Virus has capability to modify itself in such a way that can target any kind of cells. As you know that the coronavirus has certain receptors that particularly inject to the lung cells, right? Anyone know the receptor name? I think CP2 or something like that, right? So that virus has very high affinity to our lung cells. Second thing, each virus have their own genetic material inside the cells. The virus is what? Virus is just capsid protein. There is a ball. Ball is made of the capsid protein. Inside the protein there is a genetic material inside, right? In case of coronavirus, there is a RNA inside, right? Each virus contains either RNA or DNA. So in case of coronavirus, it has RNA. That's why we use the RNA vaccine. That's why we developed the RNA vaccine to inhibit the progress of RNA from the coronavirus, right? So that this is very important because of they have capability to deliver the gene of interest, whatever you want, right? Second thing is multiplication that we do not want. So we want to use the virus. We can make a binding affinity with our target cell. For example, if you are targeting the heart cell, liver cell, kidney cell, that is we can take advantage. Second thing, we can put our gene of interest into their body, which is very good. But we do not want this virus to multiply in your cells, right? So what we have to do? We have to do some. Here is the life cycle. You can see there is a multiplication part somewhere here that we do not want to amplify. Otherwise, if you amplify in your body, it's going to cause the disease. So what scientists does? They did some mutation in AV virus in such a way that it has binding affinity, it can carry your gene of interest, but it cannot multiply. So how this works? So this is AV virus, how it look like. It contains DNA, not RNA, like bacteriophage is another bacterial virus that we are not using as a tool. And another advantage of using AV is cannot cause in human disease in general. Wild type AV cannot cause any disease in human. That's why we use this virus as a tool. Okay, so if you look the genomic structure of this virus, this is how it looks like. It has a rep and cap. There is two protein inside their genetic material called rep and cap. There is a two site here is called inward tandem repeat called ITR, both sides. I will explain you what is this and why this ITR is important. So what we did, we just replace this gene with our gene of interest. For example, if any lung disease associated with the CFTR mutation in the asthma, in case of asthma, so you can have a good CFTR gene clone here on either side of ITR, right? In circular plasmid. Now you have a one plasmid that contains your gene of interest. You can clone any gene of interest. If you can clone the GFP, for example, just to see the fluorescence expression of GFP in your cell. Very simple. Now, virus also need their rep cap, right? So we clone another vector that has a rep cap. You can express it with the CMD promoter or anything like that. Now we have two plasmids. One carry your gene of interest, another carry your rep cap that virus required. This gene of interest has another side of ITR. So like I said before, we do not want to multiply the virus, so we did some mutation in the rep protein in such a way that it has binding affinity, that it can carry your gene of interest, but it cannot multiply. Right? Now we introduce third virus, third plasmid called helper plasmid. So once you transfect all three plasmid into the cell line, especially the HEK 290 293T cells, and these 3 plasmid will translate it into the protein, right? Red cap is kind of capsid protein, right? Now capsid protein has ability to start and aggregate together. This protein here is kind of assembling, right? Assembling. So during the assembly, virus need their own gene of interest as well, right? Each virus has to have their genetic material inside. So now virus will see where is my idea. This is how virus recognize their gene of interest by looking the idea. So gene therapy is saying that if you inject the AV, how many doses you have to give, right? In case of animal, 1 dose is enough.
तो वो सबसे बड़ी जो चैलेंज आता है वो यहाँ पे हम काफ़ी अच्छे तरीके से डिस्कस की, जो भी एडवांटेज है डिसएडवांटेज है, व्हाट आर द लिमिटेशंस, वी हैड डिस्कस वेरी वेल. ओके, so you all are welcome for the cup of tea. Cup of tea is waiting outside.
वीडियो देखा मैं हंसते हंसते भाग
कहाँ से आए?
तो अच्छा जो आपको से नहीं वो आपने वो बताया था मैंने आपको डायग्नोस्टिक सेंटर
लास्ट बोलना की हालचाल, ठीक है? अगर आपने देखा ना, अगर मैं बोलूँगा नमस्कार आदाब सत श्री अकाल, आपको बोलना है की हालचाल, कैसे? ठीक है, प्रैक्टिस करते हैं. नमस्कार आदाब सत श्री अकाल, ठीक है? ये प्रैक्टिस करना है, ठीक है?